Episode 11. Does ribavirin affect viral reservoirs in the gut or CNS? Welcome to another episode of Ribavirin Reminiscence and Lingering Load in Long COVID-19. I'm your host Anushka, a genomics enthusiast who spent years decoding the intricate blueprints of life's machinery. Today's question is like a labyrinth in a storm, complex yet elusive. Can ribavirin, that tenacious antiviral warrior, infiltrate the hidden viral reservoirs suspected of fueling long COVID's persistent symptoms? We are zooming in on two shadowy strongholds, the gut and the central nervous system, where SARS-CoV-2 may lurk long after its initial stage. We'll navigate the pharmacokinetics, tissue distribution, and the foggy unknowns of ribavirin's reach. Joining us is Saakshi, a brilliant mind with a master's in microbiology whose passion for unraveling microbial mysteries brings a sharp lens to this puzzle. Saakshi, welcome to the show.
Thank you, Anushka. This question is a critical frontier in antiviral drug repurposing, one that's both fascinating and frustratingly underexplored.
Let's set the stage. Imagine viral reservoirs as secret bunkers tucked away in the body's terrain where fragments of SARS-CoV-2 might be hiding, plotting their next move. Sakshi, what exactly do we mean by viral reservoirs in the context of long COVID?
It's a great metaphor, Anushka. Viral reservoirs are specific tissues or cell populations where remnants of the virus, be it RNA, protein, or even replication competent virus, persist long after the acute infection has faded. For SARS-CoV-2, we suspect several key sites such as GALT or gut-associated lymphoid tissue where the immune cells might harbor viral fragments. Another one is CNS or central nervous system, particularly the brainstem or olfactory bulb, which could explain neurological symptoms. Lymphoid tissue acting as secondary hideouts, and potentially even bone marrow, though that's less studied. These reservoirs could be driving chronic immune activation, sparking symptoms like fatigue or brain fog, even when standard PCR tests come back negative. The challenge is detecting and targeting these elusive pockets.
Like a courier navigating a sprawling city, ribavirin must deliver its antiviral payload to these distant outposts. But how well does it reach the gut and CNS? Summarizing the challenge, it's about whether ribavirin can cross the body's borders to confront the enemy. Sakshi, what does the pharmacokinetic map tell us?
Ribavirin's journey is indeed complex. It's orally bioavailable, widely distributed, and has a knack for accumulating inside cells, particularly red blood cells, which act like transport hubs. But the terrain gets tricky. Now let's look at the gut and CNS separately. Talking about the gut first, ribavirin does reach gastrointestinal tissues with animal models showing decent penetration into the mucosa, especially with sustained dosing. However, the concentration varies, and we don't know if it's enough to tackle viral remnants in the gut associated lymphoid tissue. Now, talking about CNS, here's where the road gets rocky. Ribavirin can cross the blood-brain barrier, but it's like squeezing through a narrow gate. Cerebrospinal fluid levels are typically less than 10% of plasma concentration. In acute infections like viral encephalitis, the inflammation might loosen the barrier, hence allowing more drug through. But Long COVID likely lacks that disruption, hence making CNS delivery a steep climb.
So, Ribavirin is not exactly storming the gates of either fortress with ease. The gut seems within reach, but the CNS feels like a walled off kingdom. Is that a fair read?
Spot on. It hinges on factors like dosing, formulation, and the disease's state. The gut is a more accessible outpost. The CNS is a pharmacological fortress, heavily guarded by the blood brain barrier.
If Ribavirin is struggling to reach these barriers, are there ways to re engineer its approach? Think of it like equipping our courier with a better vehicle or a secret tunnel. What strategies might enhance ribavirin's delivery to these critical sites?
There are promising ideas, though many are still in the lab or theoretical, but promising nonetheless. Let's take liposomal formulations, for example. The liposomal formulations could act like stealth vehicles, hence improving tissue specific targeting. Then there's intranasal delivery. Sounds simple, right? But it might offer a backdoor to the CNS, bypassing some of the blood brain barrier defenses. Prodrugs of ribavirin could be designed to release the active drug precisely where it's needed. And combining ribavirin with agents such as mannitol, which temporarily open the blood brain barrier, has been tested in other diseases like viral encephalitis. The catch: none of these have been rigorously explored in long COVID models. So, we are still sketching the blueprints.
Let's pivot to the evidence then, or its lack thereof. If ribavirin is our arrow, have we seen it hit the mark in these viral reservoirs? To summarize, we are searching for proof that ribavirin can strike where SARS-CoV-2 hides. Saakshi, what do the data tell us?
Frustratingly little for SARS-CoV-2 specifically. In other RNA viruses like Lassa fever and hepatitis C, ribavirin has shown it can reduce the antigen loads in tissues like the liver and also lymphoid organs. There's also some animal data suggesting ribavirin lowers the viral RNA in gut for related viruses, but the CNS, it's largely uncharted territory. No published studies showed ribavirin clearing SARS-CoV-2 or its antigens from the human brain or spinal cord. We are leaning heavily on extrapolation and hope at this point.
This feels like a high stakes treasure hunt where the prize is relief for long COVID patients. Why is nailing ribavirin's reach so critical for those grappling with symptoms like brain fog or GI distress?
It is because if those symptoms are fueled by viral remnants in specific tissues, then systemic antivirals like ribavirin must actually reach those sites to make a difference. If they are just circulating in the bloodstream, it's like sending firefighters to the wrong address. The blaze keeps burning. Effective treatment hinges on getting the drug to the root cause, not just
So,
flooding the system.
so the issue might not be ribavirin's firepower, but whether it's aimed at the right target. Am I right? Like the sharpshooter firing at shadows instead of the source.
Exactly. Ribavirin is a capable soldier, but it might be deployed to the wrong battlefield.
As we chart the path forward, what studies could illuminate this murky landscape? If ribavirin's potential is like a locked chest, what keys do we need to unlock it?
We definitely need a multi pronged approach, such as tissue biopsies from long COVID patients, gut, CSF, lymph nodes to confirm viral persistence and also measure the ribavirin levels there. Another thing is animal models of persistent SARS-CoV-2 in the brain or gut treated with ribavirin to test efficacy. Imaging techniques to track drug distribution in real time, if feasible, and most crucially, human trials that measure both clinical outcomes like symptom relief and also virological markers like antigen clearance in these tissues. It's a tall order, but these steps could bridge the gap between theory and therapy.
Saakshi, thank you for guiding us through this scientific odyssey. You have shown us that the question isn't just whether ribavirin can fight, but whether it can find its foe in the body's hidden corners.
Absolutely, Anushka. Antiviral strategy is a blend of virology, pharmacology, and relentless detective work.
So, next time on Ribavirin reminiscence and lingering load in long COVID 19, we'll dive into whether ribavirin can accelerate viral clearance through lethal mutagenesis and what that might mean for chronic conditions like long COVID. Until then, I am Anushka reminding you sometimes the problem isn't the map, it's whether the medicine reaches its destination.